Okay, so now we're going to talk about neurons at rest, because it turns out that neurons are not always carrying electrical messages. Remember that neurons are going to carry an electrical message when they detect some sort of important change, right? If we're talking about a sensory neuron, let's say a neuron in your cochlea, right, in your inner ear, it's going to respond when a sound wave arrives, right? There's got to be some reason for it to fire because it's carrying information. So most of the time, or much of the time, the neuron is actually going to be what we call at rest. And I put this in quotes on the overhead because it's actually a pretty energy-intensive process to maintain a neuron at rest. So let me just describe what we're looking at here. So here is a cell. This could be, let's say, part of the cell body of a neuron, right? Because you can see the nucleus here. If we just take a cross-section of the membrane, we just take this little piece here and we expand it out, okay? What we're going to find is we have, our typical phospholipid bilayer, right? So you can see the phospholipids, right, in a bilayer, isolating their, lipid tails that are, don't like charged substances, right? And their phosphate heads, which are oriented to the extracellular space, right, and the intracellular space, okay? And then we have a series of proteins, right, that are embedded in the membrane that will permit the passage of substances, charged substances, from one side to the other, okay? Now, in a neuron at rest, I want to go over some of the constituents, some of the substances and ions that you're going to find that are clustered around the membrane, okay? So again, phospholipid, that allows for, largely impermeable, right? It makes it difficult for charged substances to pass through, right? But you'll notice there are some channels that are here. Remember, some of the proteins that are manufactured based on instructions in your genes include what we call ion channels, right? And this is an open channel. You can see this channel doesn't have any kind of gate or door, right? It's just open to the passage of K+, which is potassium. So the potassium ion can flow from one side of the membrane to another because this protein channel, which is specific for for permitting the passage of potassium, sits here in the membrane. And I actually draw two of them here, right? There's two of them that are located. The membrane is mildly permeable to potassium, not like entirely permeable to potassium, but potassium can flow through the membrane in a neuron at rest. Some sodium can flow too, but a lot less. So this is Na+, which is the sodium ion. And this is a sodium channel, right? It doesn't have a gate or a door, so it's ungated, right? But there are very few of these. They're kind of referred to as leakage channels. They allow a little bit of sodium to leak, from one side of the cell to the other, okay? I want to identify a few other structures here. So we have phospholipid bilayer. We have some open potassium channels. We have a very few open sodium channels. We also have this squiggly looking thing down here represents a protein. This is, built up of amino acids based upon instructions, for manufacture that are found in your genes, right? And these proteins I put here on the intracellular side, right? Well, the reason is because proteins are manufactured in your cells, right? So you'll find the largest concentration of proteins on the inside of cells and not the outside. This will become significant. I write here anions, which means negatively charged, because proteins are built up of those 20 amino acids, and a majority of those 20 amino acids actually have a negative charge. So proteins tend to be more negatively charged than positively charged. So the presence of proteins in the intracellular space inside the neuron basically bias the inside of the neuron to a negative charge. So it gives it a slightly negative cast. Okay. Now, there's another structure that I haven't mentioned yet, right? But it's probably the most obvious because it's the largest, right? This is this giant thing that I'm showing here embedded in the membrane. It's a protein, again, manufactured based on instructions in your genes, right? And it's known as the sodium-potassium pump. And pump should imply energy is necessary to push something from one location to another. And if you're using energy, expending energy to push something from one location to another, it implies that you're fighting some kind of force, right, to get that thing from one side to the other. So we'll get to all these aspects in a moment, but I want you just to get a sense of what it's pushing in which direction. okay? So what it's doing is taking 3 sodium ions from the inside, the intracellular space, right, and kicking them out, pumping them out into the extracellular space. At the same time, it is pulling in 2 potassium ions and pumping them in to the intracellular space. Now this is a machine, this protein pump. It actually, I want you to imagine it as a physical kind of mechanism with like maybe some scoops on the inside, let's say 3 scoops that can hold and fit in 3 sodium ions, right? And 2 scoops on the outside, which can grab onto, 2 potassium ions. And when energy is introduced, and energy in the cell is found in the form of a chemical that's generated whenever you burn sugar in your cell. It's called ATP. It's a form of it's a chemical energy. It's found in the bond between one of these, P's here. TP stands for triphosphate, 3 phosphate molecules, or 3 phosphate ions. And by breaking that bond, or one of those bonds, you generate diphosphate, right? But you transfer the energy stored in this molecule, the chemical energy, into what's called kinetic or movement energy. This pump literally takes those scoops that are holding the sodium and takes those scoops that are holding the potassium and shifts, right? And shoves the 3 sodium out and brings the 2 potassium in. So there is a physical movement, that's literally going to be pumping sodium out of the cell. At the same time, it's bringing potassium into the cell.